Today, I would like to introduce you to the world of photosynthetic cyanobacteria and how we can unleash their power by using synthetic biology. My name is Ilka Axmann, and the Institute of Synthetic Microbiology, which I'm heading, is located at the new Center of Synthetic Life Sciences at the campus of Heinrich Heine University Düsseldorf. Synthetic biology enables us to engineer life. We are able to design and to construct systems that do not exist in nature, aiming for artificial systems with a programmable behavior. I think the most prominent examples of synthetic biology these days are the COVID-19 vaccines, the mRNA vaccines. Here, a synthetic DNA is a template for the mRNA. So you cannot derive the DNA from the virus itself because it's an RNA virus. So the DNA is synthesized, and an RNA, an mRNA, the messenger RNA of the cell, containing the information for the virus spike protein, synthesized from this artificial DNA. This mRNA can be packaged in lipid nanoparticles, again, a synthetic device, which then can be delivered to the cells to stimulate the immune response. So the great thing here is that we do not rely on natural existing sequences, on natural DNA, nor on natural existing organisms. So we purely depend on the dogma of biology, DNA transcribing to RNA, translating into protein. But when we do not start from naturally existing DNA, we are free in our designs, and we can really choose a sequence. So in terms of the COVID-19 sequence, that means we can predict mutations, we can change the DNA, the template for the vaccine, and already design new and better vaccines for this pandemic, and in a very fast time scale. So normally in the past, it took about 10 years to arrive at a successful vaccination. So these times, the vaccine was designed and produced within less than a year, which is, I think, an amazing success of synthetic biology. The principle driving synthetic biologists is that we have to create our systems piece by piece to fully understand them. So what we do not create, we do not understand. And therefore, synthetic biology highly relies on well-characterized parts. So we need defined pieces, biological pieces and synthetic pieces, to put them together for our systems. So these parts can be sequences of nucleotides with a specific function. The parts are then assembled to larger devices, and multiple devices can be plugged together to a system. So that is what we speak about as a synthetic system then. These synthetic systems have to run through an engineering design cycle, through a design, build, test, and prove cycle, which means that the initial design, which has built, has to be tested, most likely fails in the first round, then it gets improvement and runs the cycle again as long as it fulfills all the criteria matching the demands for the synthetic system. Synthetic biology allows us to engineer cyanobacteria to learn the new tricks. Besides producing naturally sugar and oxygen via their photosynthetic metabolism, we can metabolically engineer cyanobacteria to produce valuable compounds of our needs. For example, biofuels, food supplements, pharmaceuticals, and many more. Because the cyanobacteria are the progenitors of plant chloroplasts, their metabolism has a lot of similarities to plant chloroplasts. So for example, they have a naturally running non-methylmab pathway producing squalene. like in a chloroplast. So we thought about synthetically producing plant metabolites in the cyanobacteria, for example, plant terpenes. So we choose 2 different genes from Arabidopsis thaliana, from a plant, to synthetically introduce them into our cyanobacteria, in this case, Pseudocystis. So the gene in blue, which is an epoxidase, and the gene in red, which is the cyclase, were integrated into the cyanobacterial metabolism, and we checked if we were able to produce metabolites originally found in plants. And indeed, our mass spectrometry analysis showed that we have the naturally occurring squalene fully metabolized into the oxidosqualene in case we have the epoxidase present in the cyanobacteria. And further, if we have the right cyclase, in this example, the cyclase for cycloarsenol, we're even able to produce the plant sterol, cycloarsenol. So that is working nicely in the cyanobacteria, and mounts of the plant terpenes were already quite high. However, we sought to further boost the synthetic plant terpene production, and here we need a closer look into the pathway of the cyanobacteria. You see that there is a gene, CRTE, and the enzyme is transforming 3 different metabolic steps within this network. So here it's not only metabolizing FPP towards the plant terpenes, but it is also a precursor for later on producing carotenoids, which are naturally used and needed by the cyanobacteria. So to change the fluxes, the direction of these network needs balancing, because we cannot stop production of carotenoids, otherwise cyanobacteria would not survive. So we came up with the idea to downregulate the CRTE gene by the CRISPR-I technology developed in the Hudson lab in Sweden, in combination with our tunable synthetic promoters, so that we can nicely find the right level of downregulation for CRTE, that the carotenoids production is reduced, but still enough to keep the cells alive and happily growing. Additionally, we implemented a heterologous gene from a different organism into the pathway, which is ISPA, which is performing only 2 functions of CRTE, towards FPP and then further towards the plant terpenes, but not producing further carotenoids. So this combination, downregulated CRTE gene and heterologously running ISPA, enabled us to balance the metabolic fluxes in a way that the cells are alive and growing, but producing up to 5-fold more of our plant terpenes. So here we can show that cyanobacteria are indeed a successful platform which can be engineered towards the plant terpenoid biosensors. And you can easily spot by eye that we changed our host in a way that our cyanobacteria, which are normally rather green, after engineering, they now appeared as pure blue cultures, so due to the reduced level of the carotenoids. The work I presented was performed by PhD students of my lab. Anna Behle, Max Dietsch, and Anna Germann supported the work, as well as former bachelor student Pierre Sake, in cooperation with people from Forschungszentrum Jülich, Thomas Trepper and Anita Löschke, and with the help of colleagues from the Technology Institute in Sweden, Lun Yao and Paul Hudson, and with support in mass spec by Philip Westhoff, and experimental design and ideas by Dennis Dean, former postdoc of my lab. So to summarize, I presented you a modular platform for plant metabolite production, and the cyanobacteria, of course, are very modular and can be outreached for further components using synthetic biology. Furthermore, there's a high diversity of cyanobacteria strains outside. More than 8,000 species and multiple strains are waiting for their application, and many more pathways, interesting genes, and metabolites can be found outside. In the end, upscaling is interesting, but the technology is available. So companies like Photonol in Amsterdam, Alter for Future in Portugal, or Algenol in the US, having large production plants which are able to grow cyanobacteria and microalgae to a very large amount. Finally, I'm convinced that we can learn from the cyanobacteria how to fuel our planet photosynthetically. So they belong to the phytoplankton, so they are a major component of our phytoplankton, and they are able to fix up to 50% of the carbon dioxide, which is an amazing amount, and run by these very tiny microorganisms. And in a way, we can use their capabilities. I'm convinced that we find a sustainable way into our future. If you would like to learn more about the cyanobacteria, of course, you're happy to contact me, to follow us on Twitter, to sign in on our open online cyanobacteria seminar series, where the talks of cyano experts are recorded and provided to the public via YouTube. With that, I would like to thank for your attention.
Yes, thank you very much for this very interesting presentation on cyanobacteria as a modular platform for photoproduction. Professor Maria Axmann will be available for questions right after now. And as mentioned before, please use the Mentimeter for questions on these topics. Professor Axmann, you are already in the line, as I see. Perfect. The first questions that came up from the audience, but also from myself, was what are the advantages of the cyanobacteria against other organisms? Because normally the people are working on bacteria or yeast, but you're concentrating on cyanobacteria. And what do you see as an advantage in this case?
Yeah, so there are several advantages, or let's rather say differences, because here we have photosynthetic bacteria, which are able to use the energy of the sun, so to use purely the light as energy source, and which are able to fix carbon dioxide naturally. So that is, I think, a big difference compared to heterotrophic organisms. So they, of course, belong on organic compounds and on the oxygen. So it's exactly our opposite, let's say. And I think here we have to focus on, of course, especially in regards to sustainability and climate change. So I think here we have the option to use this huge resource and to make them available for biotech. So it's not that they are advantages over heterologous systems. I think they will rather be a different offer different possibilities. And further, they are also interesting for biocatalysis, as they naturally produce oxygen, which is also needed in several chemical reactions. We can then, thanks to synthetic biology, introduce into the cyanobacteria artificially, and then make use of their natural pathways.
Okay, so from this, I understand that you are focusing on carbon capture as a substrate in this case. And on the other hand, what do you see as the main challenges in cultivating, for example, the cyanobacteria and make them usable for these kinds of technologies?
Yeah, so as I introduced in my talk, so there are large-scale plants available. So there is cultivation systems around, which you can use for cyanobacteria, but also microalgae. So far, most of them are situated or located in areas where there is light, a lot of light whole day long. So rather in the southern hemisphere, so Spain, Portugal, but also Florida, or several plants in Asia. So I think here's something one has to adapt, but there are nice solutions available thanks to the LED technology. And of course, regarding temperature, you can here also start using greenhouses to have a production throughout the year, which is otherwise limited a little bit in winter time in our regions, that here light and temperature are a little bit too low to have the cyanobacteria happily growing, which prefer rather 30 degrees than minus 10. But I think that's an issue which can be solved soon. As I said, there's a big diversity in the cyanobacteria phylum, so there are even cyanos adapted to low temperatures, which then might be used in winter, for example.
Okay, brings me to the product, to the other end of the production. And so for me, the question is, do you see the opportunities more in base chemicals and fuels? Like you had mentioned some example like photanol, who are working on lactic acid and ethanol, or Phytonics is working on butanol, or do you see them more in specialities and specific molecules like you are working in?
Yeah, of course, that highly depends on the economy. So, as for all biotech companies and challenges, it highly depends on oil price. So, as soon as you have compete to a very low oil price, it's rather difficult to go for low value compounds like biofuels. So here, at the current situation, I think it makes more sense to go for more specialized products. And the triterpenes are a nice example, which can be outreached. But on the other hand, it's also the squalene, which is, for example, an adjuvant in vaccination. So here, I think we can make a point in rather specialized products. And then the advantage of the cyanobacteria is that we can rather purely produce these secondary metabolites. If you're using a plant or a substrate from a plant, you have to purify them further because you have a rather complex composition of these secondary metabolites. If we go into the cyanobacterial host, we have the advantage that we can produce only one of these components purely, or rather purely, and that, of course, it's much easier for extraction. Of course, also does not have the high costs of extraction. So here I see a clear advantage over the natural producers.
Okay, so can you name some specific applications where we can find these kinds of molecules like terpenoids you are mentioned, or also carotenoids and squalene? So to give a better understanding on in what direction it will go.
Yeah, so what is used are a lot of fragrance, so valencine or not cartoon, so these special smelling fragrances from plants. Our secondary metabolites are terpenoids. As I said, the squalene is a rather fatty substance which is used for as an adjuvant in vaccines, and there of course are classic substances purified from cyanobacteria, for example, the phycocyanin you can find in any of your foods, which is the blue color used in food industry. So that is coming from cyanobacteria, in particular from spirulina or Atrospira platensis. So here, this color should always remind you to the cyanobacteria as a biotech host.
Okay, we have some questions also from the audience. First, great talk, congratulations. Can this technology be applied to plant metabolites like peptides with multiple disulfide bonds? A very specific question.
Yeah, so disulfide bonds might become tricky because that's a prokaryotic system. So there it is mechanism is not at place. So here, of course, further engineering would be needed. But I would not exclude it. So of course, the machinery has to be integrated into the prokaryote as well. But could become possible. What is nicely running in cyanobacteria is a non-ribosomal peptide synthesis. So they are well known producing toxins, for example, or also pharmaceuticals, interesting for also cancer treatment, by their non-ribosomal peptide machinery. So they have large gene blocks working rather than a factory, adding one amino acid by the other and also cyclasing these and so on. So here engineering is also on its way, that we can use this or make it use for pharmaceuticals, for example.
Okay, are there any final terpene products that have been already produced in gram per gram cell dry weight? That's also a question from the audience.
There are, so far, rather from yeast than from the cyanobacteria, but it's possible. So it's just a question of, as I said, engineering and upscaling. So I'm, looking forward that can be also achieved in the cyanobacteria. So far, it's rather produced in yeast as a classic platform for biotech.
Okay.
So not cartoon, for example, is running, and some others, vanillin, for example. So I think that's the classic.
So you talked about the wide variety of cyanobacteria. Do you work with other cyanobacteria than the Nechocystis species?
Okay, Synechocystis is our model strain, so that's our lab bug. We are highly addicted on it because we have several or many tools and exactly know how to handle it. But we also have others. So we have been working on marine cyanobacteria. As I mentioned in my talk, there's marine silicococcus and marine prochlorococcus, which are mainly fueling the carbon cycle in the ocean. So we worked on that. And we do have also nitrogen fixing cyanobacteria. We are going to outreach them for application to become them as nitrogen fertilizers, also for agriculture applications. So that would be, I think, a big impact if we could make them platforms for atmospheric nitrogen fixation, because that is also not that common so far. And here I see also a clear advantage of cyanobacteria because they're capable of nitrogen fixation as well.
Okay, you talked about the different kinds of applications, and you also mentioned flagrants and colorants for example, food applications. What is about the consumer acceptance in this case? We will have several talks today also in this topic, and maybe we can lead already in this direction. Do you see any problems in this case, or do you don't fear anything in this direction?
So there, I'm pretty much looking forward that people will accept new foods and, of course, also new food additives, although it is not new. So the colorant, for example, is, I think, used now almost 10 years. And often it is named spirulina. So I think this has a very accepted name for food supplements in the community. Of course, if you call it cyanobacteria and bacteria, then people might a little bit skeptic in the beginning. But if you look into history, so the cyanobacteria have been used by African communities or even or South American people hundreds, even thousands of years ago. So that's a very natural source of food. So therefore, I think we can easily convince people who are a little bit skeptic until now, but I think there's a broad community which is well aware of these positive nutritional composition especially the Artospira platensis does have. So that's really a high source of protein and of course also vitamins, vitamin B12, which can be produced just by having light and carbon dioxide.